Imagine your cell has this master blueprint, right? An incredibly detailed guide for everything it does living, fixing itself, making copies.
Yeah.
That's the DNA.
The absolute core instructions. Exactly. But here's the wild part. That blueprint, it's under constant attack every single day. Thousands of times things hit it, corrupt it, maybe even wipe bits out. Things from outside, things from inside. It's a continuous barrage, really.
And if the cell can't handle this constant assault, well, it's not just about one damaged cell. It could lead to a malignant cell, cancer. That's why the cell treats DNA damage like a code red existential crisis. It's definitely the right way to think about it.
Yeah.
When we look at what damages cells, there's sort of four main targets people talk about. Mitochondria, those energy factories, cell membranes, the outer walls and internal compartments, the machinery that builds proteins, and then the DNA. Damage to the first three, sure, it can cause problems, even kill the cell. Right. But damage to the DNA, that's often the final straw. It forces the cell into this really stark choice. Either fix the blueprint perfectly and carry on, or trigger self-destruction, apoptosis, to stop something worse, like cancer, from starting. Okay, so this deep dive is really designed to help you, our listener, quickly get your head around this whole DNA damage pathway. We're going beyond just the blueprint idea to look at the actual molecular security system that deals with this threat, and we'll focus a lot on the central player, P53. People call it the guardian of the genome.
It's a good nickname for it. Now, the impact of any injury does depend a bit on the cell type, how tough it is, how adaptable. A muscle cell might shrug off some membrane damage.
Sure.
But when the DNA itself is hit, the rules change. If that surveillance system, the one P53 runs, if that fails, the cell doesn't just die. It might actually survive, but with a corrupted blueprint.
And that's the danger zone.
Yep. That's the absolute danger zone. Surviving and multiplying with damaged genes, that's how tumors can start. So understanding this pathway isn't just, academic. It's fundamental for understanding cancer, aging, a whole range of things.
Okay, let's unpack this then. Before we zoom in on P53, the guardian, we need to really understand the threats. What exactly is attacking the DNA? Let's get specific about the agents, the things hitting the DNA structure. And you mentioned external versus internal.
Yeah, the attackers are incredibly diverse. They come from the environment outside the cell, we call that exogenous, and also from just the normal everyday processes inside the cell, that's endogenous. Knowing where the attack comes from helps understand how the cell tries to fix it.
Okay, let's start outside. Environmental stuff. Radiation is probably the one everyone thinks of first, right? X-rays, sunlight.
Definitely. Ionizing radiation like X-rays or gamma rays is a big one. It damages DNA in a couple of main ways. First, it can be a direct hit. The energy particle physically smashes the DNA backbone.
Ouch. Just snaps it.
Can do, yeah. But the second way is often more common and maybe sneakier indirect damage. Ionizing radiation has enough energy to basically split water molecules inside the cell.
Wait, so it turns the water against the cell?
Pretty much. Splitting water, hydrolysis, creates these really nasty short-lived chemicals called free radicals. The main one is a hydroxyl radical, OH.
Yeah.
It's super reactive.
Oh, looking for trouble.
Exactly. It desperately needs an electron, so it just attacks the first big molecule it bumps into.
Right.
And DNA is a huge, vulnerable target, so these radicals cause widespread damage. And then there are chemical attackers, too. Chemotherapy drugs, for instance.
Ah, right. The drugs used to fight cancer.
Yeah, many of them, like alkylating agents, are designed specifically to cause DNA damage that can't be repaired. They might stick strands together or add big, bulky chemical groups. The whole point is to trigger that P53 pathway and make the cancer cells self-destruct.
It's still kind of ironic, isn't it? Using poisons that cause the very problem we're trying to stop, just because we know cancer cells are often already broken in how they handle that damage.
It is a bit ironic, yeah. But it exploits a vulnerability. Okay, so that's external stuff. What about the damage that just happens inside the cell from normal life?
Yeah, the internal threats.
This is where reactive oxygen species, or ROS, come in big time. These are mainly produced when our mitochondria make energy, using oxygen to make ATP, the cell's fuel.
The power plants of the cell.
Exactly. But that process, the electron transport chain, isn't perfectly clean. Sometimes oxygen doesn't get fully processed, and you end up with these reactive byproducts like the superoxide anion ORO.
So making energy inevitably creates these toxic side products.
Yeah.
And if they build up, they damage the cell that made them.
That's the core problem. Normally, in a healthy cell, there's a balance. The cell makes ROS, but it also has systems to neutralize them. But if the cell is stressed, maybe by toxins, maybe inflammation, maybe lack of oxygen, followed by its return, that's called ischemia reperfusion injury, then ROS production skyrockets.
Or the defenses get overwhelmed.
Or the defenses get overwhelmed. Either way, you get what's called oxidative stress. And once that happens, these free radicals start attacking everything: membranes, proteins, and critically, DNA.
So we're not just talking about like typos in the blueprint here.
Yeah.
What does this damage actually look like at the DNA level? How bad can it get? And how does the cell know if it's a minor scratch versus a total disaster?
That difference is absolutely key. It dictates the whole response. Oxidative stress chemical attacks, they cause different kinds of lesions. The most common ones are probably single-strand breaks, or SSBs, just one of the two DNA strands gets broken.
Right.
These happen all the time, thousands per cell per day, maybe, but they're generally seen as relatively easy fixes. Why? Because the other strand is still intact. It acts like a template. The cell uses that template to patch up the broken strand, mostly using a system called base excision repair, or BER.
So SSBs are like routine potholes. Fixable. What's the 5-alarm fire then?
That would be the double-strand break, or DSB. This is when both strands of the DNA helix get cut right across, often close together. High-energy radiation is notorious for this, or really severe oxidative stress.
Uh-oh. No template left now.
Exactly, no template. So the repair is much trickier and much riskier. The cell has a couple of options, but they're not ideal. One is called non-homologous end joining, NHEJ. It basically just glues the broken ends back together. Fast, but often messy bases can get lost or mixed up.
Error-prone.
Very error-prone. The other main option is homology-directed repair, HDR. This one is accurate because it uses the undamaged sister copy of the chromosome as a template, but it's only available during certain parts of the cell cycle, basically after the DNA has already been copied but before the cell divides.
So timing is critical for the good repair pathway.
Absolutely. And even a single unrepaired DSB, that can be enough to kill the cell. Or worse, if it gets repaired badly, it can lead to big chromosomal changes, bits swapping places, which is a major driver of cancer development.
Wow. Just the fact we're sitting here talking means our cells are constantly winning this repair battle against incredible odds. And you mentioned aging earlier. Spontaneous damage is just part of getting older.
Yeah, there's a direct chemical link too. One thing that just happens spontaneously chemically inside our cells is something called deamination. Specifically, a DNA base called cytosine can spontaneously lose an amino group and turn into another base, uracil.
Okay, so cytosine turns into uracil. Why is that bad?
Well, cytosine is supposed to pair up with guanine, C with G, but uracil normally pairs with adenine, U with A. Uracil doesn't belong in DNA normally, it belongs in RNA. So when the cell copies its DNA, if there's a uracil sitting where a cytosine should be, the replication machinery mistakenly puts an adenine opposite it.
Ah, I see. So the next copy is wrong.
Exactly. What started as a CG pair ends up becoming a TA pair in the daughter DNA strand, since thymine, T, is used instead of uracil in DNA. It's a permanent mutation caused by simple chemical instability.
A sort of internal ticking clock generating random errors over time. That's a good way to put it. And over decades, these kinds of spontaneous changes, plus maybe some unrepaired oxidative damage, they accumulate. Eventually, the repair systems might get overwhelmed, or the cell just has too much damage. This contributes to cellular senescence, where cells basically retire and stop dividing. It's one of the key links between DNA damage and the overall aging process. Okay, this is getting really interesting. So we've got the threats, radiation causing those nasty DSBs, ROS causing all sorts of damage, even to spontaneous chemical decay like cytosine deamination. Now, the crucial part, how does the cell know damage has happened? How does it detect it? And what are the immediate first steps to stop things getting worse, like accidentally copying the broken DNA? How does P53 actually get the signal?
Right, this is where the signaling cascade kicks in. It's really elegant. The damage itself doesn't usually activate P53 directly. Instead, the damage activates specific sensor enzymes, often called checkpoint kinases. Yeah. The two big players here are called ATM and ATR.
ATM and ATR, like the first responders on the scene.
Exactly like that. They're the detectors. When a double-strand break happens, ATM is usually the one that senses those broken DNA ends and gets activated. For single-strand breaks, or if the DNA replication process itself gets stuck because of damage, ATR tends to be the main sensor.
So different sensors for different types of trouble.
Pretty much. Once ATM or ATR are switched on, they act fast. Their job is to phosphorylate other proteins, basically, sticking a phosphate group onto them, which acts like an on switch or a signal flag. And one of their most critical targets for phosphorylation is the p53 protein itself.
Okay, so ATM and ATR detect the damage, then they tag p53 to tell it, hey, wake up, there's a problem. But if p53 is so important, why isn't it just active all the time? Why does it need activating?
That's a really good question. It highlights how tightly regulated this whole system is. In a normal, healthy, unstressed cell, p53 levels are actually kept incredibly low. There's another protein called MDM2, whose main job is to grab onto p53.
MDM2, the p53 handler.
Kind of like its jailer, actually. MDM2 attaches little tags called ubiquitin to p53. And those ubiquitin tags are basically a signal saying, destroy this protein. They send p53 to the cell's recycling center, the proteasome, where it gets chopped up.
So MDM2 is constantly destroying p53 in healthy cells.
Constantly. It ensures p53 doesn't interfere with normal cell growth and division when there's no crisis. It's a very rapid turnover.
Like a dead man's switch that needs to be actively held off.
That's a great analogy, but when ATM or ATR detect damage and phosphorylate p53, that phosphorylation changes p53's shape slightly. And critically, it stops MDM2 from being able to bind to it effectively.
Oh, the phosphorylation breaks the chain, sets p53 free.
Exactly. p53 is no longer being tagged for destruction, so its levels shoot up rapidly inside the nucleus. And once you have enough p53 accumulated, it can start doing its job, which is acting as a transcription factor.
Meaning it turns other genes on?
Turns specific genes on or off. It binds to DNA and controls the expression of a whole network of genes involved in the damage response. Okay, so p53's active levels are high. What's job number one? You said it buys time. Phase 1, cell cycle arrest. Absolutely. First things first, hit the brakes, stop the cell cycle. p53 primarily arrests the cell in the G1 phase, that's the growth phase, before the cell commits to copying its DNA in the S phase. You absolutely cannot risk replicating damaged DNA.
Makes sense. How does p53 physically stop the cell cycle?
It does this mainly by turning on the gene for another protein called p21.
p21, the enforcer of the pause.
That's exactly what it is. p21 is a powerful inhibitor of enzymes called cyclin-dependent kinases, or CDKs. Think of CDKs, along with their partner proteins called cyclins, as the engines driving the cell cycle forward. To get from G1 into S phase, you need specific cyclin-CDK pairs to be active, like cyclin-ECDK2 and cyclin-DCDK4. What p21 does is it binds directly to these cyclin-CDK complexes and shuts them down, puts the brakes on those engines. Without active CDKs, the cell simply cannot progress into S phase. It's stuck in G1. So p53 activates p21, p21 jams the CDK engines, cell cycle halts in G1, and that pause, that's crucial because it creates a window for phase 2, the repair attempt. Precisely. That G1 arrest enforced by p21 buys the cell precious time. During this window, the cell's DNA repair machinery gets mobilized and goes to work on the damage. p53 actually helps here too, by turning on some genes involved in the repair process itself, like one called GADD45.
So p53 both pauses the cycle and helps call in the repair crews.
It coordinates the whole initial response. The priority now is fixing those single-strand breaks using BER, maybe dealing with chemical adducts using other pathways like nucleotide excision repair, NER, trying to mend any double-strand breaks. The goal is perfect, high-fidelity repair, restore the blueprint to its original state.
But what if the repair crews can't do it? What if the damage is just too bad? Too many double-strand breaks, maybe, or the damage is too complex. How does p53 know when to give up on repair? When does that G1 arrest window close?
That's a really fundamental question, and honestly, the precise mechanisms are still being worked out, but the thinking is it relates to the persistence of the damage signal. If the damage is repaired quickly, ATM and ATR are shut off, p53 stops being phosphorylated, MDM2 grabs it again, levels drop, p21 goes away, and the cell cycle restarts.
Repair successful, release the brace.
Right. But if the damage is severe and irreparable, those 8-matter sensors stay active. They keep phosphorylating p53. The damage signals just keep screaming. And if that damage on signal persists beyond a certain threshold or for a certain duration, p53 seems to undergo a kind of functional switch.
A change in mission.
Exactly. It starts to change which genes it preferentially activates. It shifts away from promoting cell cycle arrest and repair and towards promoting the final solution.
Apoptosis. Programmed cell death. p53 goes from repair coordinator to executioner. If the blueprint cannot be fixed, p53 makes the call. This cell is too dangerous to live. It triggers apoptosis, primarily through the intrinsic or mitochondrial pathway. To do this, p53 turns on powerful pro-apoptotic genes. Two really key ones are BX and PUMA.
BMX and PUMA, the death signal activators.
They are. PMA stands for p53 upregulated modulator of apoptosis. It's a particularly strong killer activated by p53.
So how do BX and PUMA actually, kill the cell? What do they do?
They both work mainly at the mitochondria. BAX, along with a related protein called BAK, can basically punch holes in the outer mitochondrial membrane. PMA helps this process by neutralizing the cell's own protective anti-apoptotic proteins, like BCL2, which normally keep BX and BAK in check.
So PUMA clears the way for BX to poke holes in the mitochondria.
Essentially, yes. Once those pores form in the mitochondrial membrane, stuff starts leaking out that shouldn't be in the main part of the cell, the cytosol. The most critical thing that leaks out is cytochrome C.
Cytochrome C, isn't that usually involved in energy production?
It is inside the mitochondria. But when it leaks into the cytosol, it acts as a lethal signal. It binds to other proteins to form a complex called the apoptosis, which then activates a cascade of enzymes called caspases.
The caspase cascade?
Right. Think of caspases as the demolition crew. They are proteases, enzymes that chop up other proteins. Once activated, they systematically dismantle the cell from the inside, chopping up structural proteins, degrading DNA, shutting down essential processes, leading to the cell's neat and tidy self-destruction.
It sounds incredibly orderly, like a very disciplined sacrifice. Why go to such extreme lengths? Why not just limp along with a few DNA errors?
Because that's the essence of p53's role as a tumor suppressor. It's making what you could call a cellular ethical choice. The cell chooses to die rather than risk surviving with damaged DNA. Why? Because a cell that survives with significant mutations, especially things like unrepaired double-strand breaks, has the potential to become cancerous.
Right, it could start dividing uncontrollably.
Exactly. By triggering apoptosis, p53 eliminates that threat cleanly. It sacrifices the individual damaged cell to protect the health of the whole tissue, the whole organism. It prevents the propagation of potentially cancer-causing mutations.
Which brings us, inevitably, to the dark side. What happens when the guardian itself, p53, is compromised? What if it's mutated or missing?
The consequences are profound. Mutations in the gene for p53, called TP53, are found in something like over half of all human cancers. It's the single most commonly mutated gene in cancer.
Wow, more than half.
More than half. And if a cell has a broken or missing p53 protein, the whole system falls apart. The cell might still detect the DNA damage ATM and ATR might still get activated, but the signal stops there.
The message doesn't get through to the command center.
Right. Without functional p53, the cell can't effectively turn on p21. So it can't properly arrest the cell cycle in G1.
It blows right through the checkpoint.
It blows through the checkpoint, trying to replicate damaged DNA. And crucially, without functional p53, it also can't effectively turn on BAX and PMA, so it can't trigger apoptosis even when the damage is lethal.
So the alarm is blaring, but the security guard is gone, or maybe even helping the intruders. The cell just ignores catastrophic damage and keeps dividing, copying the errors.
Precisely. That failure to arrest, failure to die in the face of lethal damage is a classic hallmark of cancer. Cells with mutant p53 just keep proliferating, accumulating more and more mutations because the checkpoints are down. This leads to genetic instability, which fuels tumor growth and evolution. It's also why cancers with p53 mutations are often harder to treat with traditional chemo or radiation, because those therapies often rely on triggering p53-dependent apoptosis.
That makes so much sense. Okay, let's just take a step back for a second and connect some dots. We talked earlier about oxidative stress, those ROS, being a major internal source of DNA damage. It's interesting how the cell has defenses before p53 even gets involved, trying to stop ROS from reaching the DNA in the first place. It connects membrane health and DNA defense.
Absolutely interconnected. Because free radicals, like that hydroxyl radical, are so reactive and mobile, the cell needs layers of defense. Think of the antioxidant systems as the outer walls, the first line of defense trying to neutralize the enemy before it even gets near the castle keep, which is the nucleus containing the DNA.
So there are specific enzymes dedicated to just mopping up these ROS. What are they?
Yes, several key ones, often located right near where ROS are produced, like the mitochondria and other organelles called peroxisomes. The first line against the superoxide anion are the superoxide dismutases, or SODs.
SODs, what do they do to superoxide?
They convert it very quickly into hydrogen peroxide. Now, hydrogen peroxide is still an ROS, still potentially damaging, but it's significantly less reactive than the superoxide anion.
Okay, so SOD turns a really bad guy into a slightly less bad guy. Then what happens to the hydrogen peroxide?
Then the second line of enzymatic defense kicks in. There are two main players here. One is catalase. It's found mostly in peroxisomes, and it's incredibly efficient. It breaks down hydrogen peroxide directly into harmless water and oxygen. Simple, clean disposal.
Nice. And the other one?
The other major enzyme is glutathione peroxidase, or GPX. This one is found in the cytosol and mitochondria. It uses a small antioxidant molecule called glutathione as a helper to break down hydrogen peroxide, also ultimately producing water. It also helps neutralize other harmful peroxide molecules.
Okay, SOD, catalase, GPX. Sounds like a pretty robust system.
Whoa.
If the cell can turn superoxide into hydrogen peroxide and then instantly turn that into water, why is oxidative stress still such a huge problem in so many diseases and aging? Why doesn't the system always work?
It comes down to balance and capacity. If ROS production just overwhelms these enzymes, maybe during intense inflammation or exposure to certain toxins or that ischemia reperfusion thing we mentioned, the system gets saturated, hydrogen peroxide levels can build up.
And that's bad because hydrogen peroxide, even though it's less reactive than superoxide, can react with transition metals that are naturally present in the cell, like iron FeO. This reaction is called the Fenton reaction, and the result of the Fenton reaction is the creation of the absolute worst ROS of all, the hydroxyl radical. The hydroxyl radical again, the one that causes the double strand breaks.
That's the one. It's incredibly reactive, and unlike superoxide or hydrogen peroxide, the cell has basically no enzymatic way to directly neutralize it. Once it's formed, it reacts instantly with the first thing it hits. If the cell's other antioxidant defenses, things like vitamin E and membranes, vitamin C, glutathione itself, were also overwhelmed, that hydroxyl radical can reach the DNA.
And boom! Direct hit, potentially a DSB. And now we're back to activating 8-MADR and the whole p53 pathway. The failure of the outer defenses leads directly to the crisis at the core.
Exactly. The systems are layered, but a severe enough oxidative insult can breach them all and force the p53 decision.
Okay, it's crystal clear just how central p53 is. Guardian, checkpoint, executioner. Its failure is catastrophic, leading to cancer. Given that, what's happening on the cutting edge? How are scientists and doctors trying to use this knowledge today? Are there new ways to manipulate this pathway or other repair systems?
Yeah, the field has definitely moved into a more sophisticated phase. It's not just about broadly causing damage anymore. Now it's about precision, often guided by knowing the specific status of p53 in a patient's tumor. Because in many aggressive cancers, p53 isn't missing, it's just mutated, often just a single amino acid change that messes up its structure and stops it working properly.
So the protein is still there, just broken. So the cutting edge is trying to fix the broken guardian, not just bypass it.
That's a major strategy, yes. It's called p53 reactivation. Researchers are developing small molecule drugs that are designed to physically bind to these common mutant forms of p53 protein.
Like a splint for the broken protein?
Kind of like that, yeah. The idea is the drug helps the mutant p53 refold into something that looks and acts more like the normal, functional, wild-type protein. If they can achieve that, even temporarily.
Then the reactivated p53 could suddenly turn on p21, arrest the cell cycle, and maybe even turn on BAX and PUMA.
And push that tumor cell into apoptosis.
Yeah.
It's about turning the tumor's own faulty protein against itself. There are drugs in clinical trials aiming to do just this. It's a really exciting area.
Wow, trying to rehabilitate the corrupted guardian. That's clever. What about other approaches? You hear about synthetic lethality. How does that relate to DNA repair and p53? Synthetic lethality has been a huge success story, especially in cancers with faulty DNA repair pathways, which often includes those with p53 mutations. The basic idea is this. Cancer cells are often defective in one major repair pathway, like say the one involving p53 and downstream responses. Because of that defect, they become hyper-dependent on other backup repair pathways just to survive the baseline level of DNA damage they constantly experience. Okay, so they've lost plan A, and now plan B is absolutely critical for them. Precisely. So, synthetic lethality says, what if we deliberately inhibit plan B? In a normal cell that still has plan A working fine, inhibiting plan B might not be a big deal. It can cope. But in the cancer cell that relies only on plan B, inhibiting it is lethal. You knock out their only remaining crutch.
Exactly. The most famous example involves enzymes called PARP inhibitors. PARP enzymes are really important for fixing single-strand breaks, mainly through that base excision repair, BER pathway we talked about.
Okay, PRP fixes SSBs. How does blocking PRP kill a cancer cell, especially one with bad P53?
Right, think it through. In a normal cell, if you block PRP, those SSBs might hang around a bit longer. But the normal cell has functional P53. It can sense the problem, pause the cell cycle with P21, and give other backup systems time to fix the SSBs, or maybe trigger apoptosis if needed. It survives.
Okay.
But now take a cancer cell, maybe one with mutant P53, or perhaps a defect in another pathway like homology-directed repair, often seen in BRCA mutant cancers.
Yeah.
You inhibit PRP in this cell. Now, the SSBs can't get fixed efficiently by BER.
And the cell doesn't pause properly because P53 isn't working.
Exactly. So the cell blunders into S phase, tries to replicate its DNA with all these unrepaired SSBs still present. What happens when the replication machinery hits an SSB?
It crashes. It crashes. The replication port collapses, and that SSB gets converted into something much worse. Double-strand break. So blocking the SSB repair leads to DSBs.
Loads of them. And because this cancer cell often has defects in DSB repair too, like faulty HDR and BRCA cancers, and because its P53 can't trigger apoptosis effectively, the sheer number of DSBs becomes overwhelming. The genome shatters. The cell dies.
While the normal cells, which could pause or had better DSB repair, tolerate the PRP inhibitor much better.
Precisely. It's synthetic lethality. You're exploiting a specific vulnerability created by the cancer's existing defects. PRP inhibitors have been a major breakthrough, especially for certain types of ovarian and breast cancer linked to BRCA mutations.
That's a brilliant example of targeting the cancer's weaknesses. Okay, so we see huge progress in cancer. Is this deep understanding of DNA damage and repair moving into other areas too, like neurodegenerative diseases or aging itself?
Oh, absolutely. The link between the accumulation of DNA damage and many aspects of aging, as well as specific chronic diseases like neurodegeneration, is a really hot area of research. We actually get strong clues from rare genetic syndromes. There are several inherited diseases where the core problem is a defect in a specific DNA repair machine.
Like what? What happens if you inherit faulty repair genes?
Well, often these syndromes lead to symptoms that look like dramatically accelerated aging. For example, take pathways involved in fixing bulky damage, like the kind caused by UV light from the sun. That system is called nucleotide excision repair, or NER.
Okay.
Rare diseases like Cockayne syndrome or xeroderma pigmentosum are caused by mutations in NER genes. And patients don't just get severe sunburns or skin cancer risk. They often have severe developmental problems, neurological degeneration, and features of aging appearing decades early.
Wow. So failing to fix UV damage essentially can make your brain age faster.
It strongly suggests that maintaining DNA integrity is crucial for long-term health, especially in cells that don't divide much, like neurons. Neurons have to last your entire lifetime. They can't just be replaced easily. So if they accumulate DNA damage they can't fix, it leads to dysfunction and eventually cell death, contributing to neurodegenerative diseases.
That really raises the stakes. What about the sensors? You mentioned ATM earlier, the DSB sensor. What if that's broken?
Another key example. Mutations in the ATM gene cause a disease called ataxia telangiectasia, or AT. These patients have extreme sensitivity to radiation, problems with movement control, ataxia, weakened immune systems, and a very high risk of cancers like leukemia and lymphoma.
Because the very first step in detecting the most dangerous DNA damage is faulty.
Exactly. The whole P53 dependent response pathway is crippled from this start. It really underscores how fundamental this surveillance system is. The fight against aging, against chronic inflammation, against neurodegeneration. It seems increasingly clear that it's deeply intertwined with the fight to maintain DNA integrity through efficient repair and effective surveillance systems like P53. That really brings everything together. Okay, let's try and quickly recap this amazing journey. We started with this core idea. Our DNA, the blueprint, is constantly under siege. Radiation, toxins from outside, reactive oxygen species from inside, even just spontaneous chemical decay, a constant battle. And we saw how the cell responds with this incredibly sophisticated security system centered on P53. Damage sensors like ATM and ATR activate P53.
Right. And activated P53 first hits the pause button on the cell cycle using P21 to create a window for the repair crews to work.
And if the repair succeeds, great. Breaks off, cycle continues. But if the damage is too severe, P53 switches roles. It activates the killer genes, BAX and PMA, triggering apoptosis via the mitochondria and the caspase cascade.
The honorable sacrifice to prevent potential cancer. We also touched on the outer defenses, SOD, catalase, GPX, trying to stop ROS before they even cause the damage.
And how failure there can lead to those deadly hydroxyl radicals. And finally, we looked at how medicine is getting smarter. developing p53 reactivators, or using synthetic lethality with PRRP inhibitors to exploit the specific weaknesses of cancer cells.
So when you boil it all down, what does this really mean for us? It sounds like the fundamental processes driving disease, driving aging. They're tied directly to how well our cells protect and repair their DNA. The health of our entire body seems to hinge on this molecular checkpoint working perfectly.
It really seems that way. We spent a lot of time talking about the bad news, how mutated p53 leads to cancer. But let's finish with the provocative thought, the flip side. Imagine if research gets really good at boosting our natural DNA repair systems.
Enhancing the repair crews.
Maybe making pathways like base excision repair or NER even more efficient, and ensuring p53 always works perfectly, always initiating repair when possible, always triggering apoptosis when necessary. If we could guarantee that every cell maintains a near-perfect blueprint throughout life, could we dramatically slow down aging? Could we prevent age-related diseases by tackling the root cause, the accumulated damage?
Are we essentially talking about eliminating senescence by ensuring perfect DNA fidelity?
It certainly raises that fundamental question, doesn't it?
Yeah.
One of the real biological limits if we could perfect this ultimate cellular security system. Something definitely worth thinking about.
Indeed it is. Well, thank you for joining us on this deep dive into the cell's absolutely critical, incredibly complex DNA defense network.